Now, before starting our solving our problems in Excel, we first have to make sure that our Excel version is updated for our solving for today. So firstly, to check if your Excel was made for our problem solving for today, first go to the option, there you go, and then you would see this add-ins. So click on it, and then you would see this manage Excel add-ins. In here you would see the analyst toolpack, the analyst toolpack VBA, and our solver add-in. So all of it has to be checked to be able to solve the problem that we are going to be solving for today. So if you checked all of these three, the analyst toolpack, the analyst toolpack VBA, and our solver add-in, press the OK, then you're good to go to solve the problem. So good day everyone. Today I'm gonna be talking about and also gonna be teaching you to how to graph our regression analysis problem in Excel. So for our first problem, we have the leaf size and chlorophyll content. So our problem in here would be, we would investigate whether the leaves that are larger, that was indicated by their surface area, tends to higher concentration of chlorophyll. So firstly, after observing the problem and then analyzing it, today we are gonna go graph the problem. So firstly, we're gonna input the data and also the information in the problem 1 chart. So we have in here the observation column and then the leaf surface area, which is the X, and its measurement is in centimeters squared. And then for our Y is the chlorophyll content, which is in milligram or grams. And now we're gonna input the text and also the data to our first problem. And then now, after we're done putting the text and also the data for our first problem, now we're all gonna go select the datas starting from the B3 in here all the way to D12 there. First, click on this cell B3 and then drag it until to D12 there. We have all selected the data, and then now let's go to insert, and then we have this recommended chart found. Click on it and then you would see this scattered chart. So click the OK. So yeah. So this is our scattered graph. Now we are gonna go and solve the person of our first problem. So first type equals to any cell. So I pick the F4 cell. Go. So first type the equal sign and then the person and then open parenthesis, then here you could pick your first ray, which is the cell C3 up until to C12. There you go. There. So C3 up until to C12. Then next type the comma. There you go. And then we are going to pick the next column, which is the D3 up until to D12. Drag it. D3 up until to D12, and then type out the close parentheses and then click the enter to get the answer. So this is our person for our first problem. And now let's solve for the correlation of our first problem. Firstly, we're gonna create the column of our correlation. So let's put correlation here. Then here I'm gonna input the leaf surface area. The next is for the chlorophyll content. There you go. So first thing first, you're gonna go select the cell which is the next to the leaf surface area, and then you're gonna click this, the insert function, which is this one. There you go. It will automatically put equal sign there, and then there you go. Ito dito may correlation, click it, and then okay. Then for the first array, pick the observation column. There you go. Then press the OK. And then for the second array, we are now going to select the column of the leaf surface area. There you go. Drag it until the end, and then enter, and then there you have it. And then now press the OK. And now that is the correlation of our leaf surface area. And then moving on to the chlorophyll content. And again, we are going to press the insert function and the correlation. Okay. Again, for the array 1, we are going to select the column of observation. Then enter. Then for the array 2 is the chlorophyll content. So starting from D3 up until to D12. And then enter. Then to get the answer, press the OK. Then there you have it. That is our correlation for our leaf surface area, and then for the chlorophyll content. Now, for our problem 2 would be the drug dosage and bacterial growth inhibition. So in here, our problem would explore the relationship between the amount of an antibiotic administered and the size of the zone of inhibition observed in a bacterial culture. Moving on for our problem 2, which is the drug dosage and the bacterial growth inhibition. So in here, we're gonna go insert our data and again our information. Once again, we have the first column observe, and then for our second column, we have the drug dosage, which is the X. Next, we have inhibition diameter for our third column, which would act as our Y. So let's zoom this a little bit. After putting our data to our column, and now we're gonna go select the C3 cell, which is this, up until to D12 in here. So firstly, click the C3 cell and then drag it along until the D12. And then we're gonna go click insert, and then again, you would press on the recommended chart. There you go. There you go. So in here you would see the linear line, and then spell out the person, then open parentheses, and then you need to pick the first column starting from C3 up until to C12. Then follow it with the comma, and then for the next column starting from D3 up until to D12. There you go. And then close it with the close parenthesis, and then press the enter to get the answer. There you have it. That is our person for our second problem. And then next, we are still gonna be finding the correlation for our second problem. And again, we are going to make a column for it. So correlation. And then here we have After inputting our information, now pick the cell next to the drug dosage to find its correlation. So again, we're going to click on the insert function. There you go. Then correlation, okay. For the first array 1, click this one to pick this observation column. Then press enter. Then for the second array, we are going to pick the drug dosage column, starting from C3 up until to D12. Then press the OK to get the answer. Then for the inhibition diameter, we are again gonna press the insert function, correlation, okay. First array for the observation column, enter. For the second array is the inhibition diameter. There you go. Press the OK. Then there you have it. This is our value for our inhibition diameter. The problem would be the study hours and the test score. So in here, in our problem, this would model the effect of the time spent studying on the score obtained in a biology exam of the students. And then moving on, for our problem 3, we have the study hours and the test score, and it is regression. So in here, we again would have our first column for observation. Second column that would act as our X is the study hours, and then the test scores for our Y. And then now we're gonna go put on our data and our information. And after putting our data, now we are once again gonna select all of the data in every cell. So starting from B3 up until to D12 in here. So click the B3 cell and then drag it along to D12. There you go. And then click the insert and then the recommended charts. You would see the scattered chart once again. There you go. Now this is our graph for our problem 3. Now for our third problem, we are going to find our person. So first, type out the equal sign and then spell out the person. And then open parentheses and then pick your first column starting from C3 up until to C12. And then follow it with a comma. And then for our next column, which is D3, until D12. There you go. And then close it with a close parenthesis. Then press enter to get the value of our person for our third problem. There you go. This is our person for our third problem. And then now we are going to find the regression of our third problem. So first thing first, you need to go to data located in the upper left corner in here, and then you would go to the analyze section. In here you would see the data analysis. Click on it and then scroll down a little bit and then you would see the regression. Click on it and then press OK, and then this would pop up. So for the input Y range, this is our Y range, which is the test score. And then for the input X range would be our study hours. So click this to select the cells. So for our Y until D3 until D12, and then enter. For our X range, C3 until C12. There you go, and then press enter. Then there you have it. And then in here you would see the output option. Click on the output range and then click on this again, so we would be able to pick in what cells will the result or the regression would be written. Here you go. So I pick in here, for example, so F7 up until to M18. There you go. And then press enter. Then okay. There you have it. So we would be now we have the summary output, then the ANOVA, and also our intercept and y(x) variable of our problem. Lastly, for our fourth problem would be the water temperature and oxygen solubility. So in here, our problem would use regression to be able to model how the temperature of water affects the amount of dissolved oxygen it can hold or its solubility. For our last and our fourth problem would be the water temperature and the oxygen solubility in regression. So again, for our first column, we have the observation. For the second one is the temperature in degrees Celsius that would act as our X. And then for our third column would be the dissolved oxygen in milligrams or liter that would act as our Y. So now, let's put on our data and information to our Excel. And now after putting our data to our chart, we are now gonna go select all of our data starting from B3 in here up until to D12 in here. Now click on the B3 cell and then we would drag it along to D12. There you go. And then click the insert and then the recommended charts. There you go. You would see the scattered chart. Press OK. There you go. Here. Now we see the linear of how the pinpoint of our data is. So earlier you see that all of our pinpoint data was going upward, right? Starting from the lower part or in the middle part and going upward, going up. Our first problem, our second problem, and then our third problem. But to our last problem, it started from the very high point and then decreasing to lower point. That is because our B value was negative. So our value in here, the A value was positive and then the B value is negative. Unlike to our first, second, and our third problem, both of their value A and B is positive. That is why their linear or their pinpoint of the data was going upward. This is the linear line of our problem 4. And our last and fourth problem, to get the person, type out the equal sign, then spelled out the person, open parentheses, and then pick your first column starting from C3 up until to C12, and then follow it with a comma and the next column for the D3 to D12, and then close it with a parenthesis and press the enter to get the answer. So here we got the negative 0.98729. And now lastly, for our fourth problem, we are going to find the regression of our problem. So once again, just like what we did before, we are gonna go and select cell here. We are gonna go to the data here and then you would go to the data analysis in here, then pick regression, then this would pop up. So again, for our Y range, it would be our dissolved oxygen, and then for our X range would be our temperature. So click this to select these cells. For our Y range up until 2D12, you have it, and then enter. And then for our X range, C3 until C12, enter. And then click on the output range, there you go. Click on the output range, and then there you go. Click this to be able to select in what part or cells will be the answer would be written. And then after selecting the cells, press the enter, and then OK, and there you have it. We once again have our summary output, our ANOVA, and then our intercept and X variable, and then here you would notice our X variable is in negative, so that is why our graph was descending downward. So there you have it. And now that I'm done portraying and also teaching on how to graph those regression analysis problem, I hope that this video helped, and I'm the presenter and the reporter, Mamalayan Hannah Renee from BS Biology 1B from LSPU Santa Cruz, presented to Mr. Victor Estalila, our professor for GEC104.